and then
Okay.
go from there. So how is oh my God, we are slammed with storms, one after another.
Yeah, that's crazy. I can't believe, I think they're saying a record number of storms that are, coming through from the slope there. So you just catch a break, right?
Yeah. What I like about is your background. The background it looks like as if you're sitting, but it's in your lab or in your office, right?
Yeah, this is the image right outside my lab. So this is between the Mayo Medical School building and the Guggenheim building where my lab is. So and I could say this is obviously a virtual background, but today it's 80 degrees and sunny here, which is odd for a later September date.
Yeah. But anyway. All right.
I see that we're starting to have panel or attendees starting to show up too. So that's good.
All right, guys, we're going to get started in about 2 minutes. I have us live streaming on Instagram to about 25,000 people and live recording on YouTube on both of our YouTube channels. So I think Randy will be on shortly and we can get started at 4 on the dot.
Okay.
If Randy's okay with that.
Yeah. So Nitesh, tell me, where are you based?
I'm a chief resident here at Rutgers in neurosurgery, and just finishing up and then heading to Miami for a fellowship with Dr. Komatar.
Oh, good. So that's the adult world, right? You're in the adult
world? Yeah.
Okay, right.
I pulled Nitesh in because I'm moderately tech savvy, but I figured a younger generation might be more tech savvy. And also, I work at 2 hospitals and I just needed a hand. It gets a little bit cumbersome when you're dealing with not just the programming of it, but also trying to run a practice post-COVID. So, or 2 practices, I guess. So, but things have been great so far. And actually, Nitesh kind of pioneered what we're doing now, which is these live feeds. So you can watch it live on Zoom, but like he said, right now you're broadcasting live to the Lenox Hill Instagram account, which is about 25,000 followers, I think. And routinely what'll happen is we'll get about, somewhere between 400 and 600 clicks. And then that'll actually dwindle down to about, somewhere between 100 and 200 steady listeners or so. So yeah, and then YouTube live streams as well. And then the videos get deposited there. But why don't we get started? Jason's running a little bit late, I guess. Hi, everyone. Welcome back. It's another edition of Tumor Talk. Here's Jason now. This is a collaborative effort between Lenox Hill Neurosurgery and the Journal of Neuro-Oncology, where we highlight recent publications in the journal that have to do with brain and spinal cord tumors. Typically, we discuss kind of, articles that have been recently published. We discuss the rationale behind them, the results, as well as the relevance. Today's a special day where we're delighted to have Dr. Khatua and Dr. Daniels speaking with us about an upcoming issue, a special issue of the journal about pediatric oncology, neuro-oncology. And Jason, did you want to say anything more about that before we kick off?
Well, first I want to commend them for helping to put together this special issue. It's an important part of the Journal of Neuro-Oncology that topics of high interest and where there's been appreciable changes in the field are oftentimes the focus of a special issue. And I think pediatric neuro-oncology is one of those areas in neuro-oncology that's changed a lot, and getting experts not just to help to edit that special issue as we have, but to contribute. I'm very pleased. I mean, this special issue, as you'll hear about, is really, I think, a high impact set of works that will, and I encourage everyone to read it and to go through those amazing contributions. But with that, I will let them speak a little bit more about the work so far.
Great. Dr. Khatua, do you want to introduce it? I know you were heavily involved in
the so first of all, thank you. This is the first time we are coming into this webinar, and we hope we will participate more. I want to thank, first of all, Dr. Shia for giving us the opportunity to bring in some leaders across the country and beyond to contribute to this special edition. So what I'm going to do in the first 3-4 minutes is just talk about the contributors and they're very briefly about them, and then allow Dr. Daniels to take over. So I think one of the first things is this is a special edition of epigenetics, basically in pediatric tumors. As you know, all that a lot of things are going on in the epigenetic machinery of pediatric tumors, different types of brain tumors, because the genomic landscape is rather silent. So we decided this would be a good opportunity to focus on epigenetics of various pediatric brain tumors. We are actually privileged to have Dr. Roger Packer, who is one of the global leaders in neuro-oncology, to give an update or an editorial where he's going to span out over the last 20 years how we have moved from the world of clinical care to the world of genomics and now into the world of epigenetics. So this is going to be a nice summary of his. Then if we go on to the gliomas, which constitute about 60% of brain tumors, we have the low-grade gliomas. And it's very interesting, over the last 5-7 years, we have really found a lot of interesting data on the various molecular impact and the subsequent into the epigenetics of low-grade gliomas. This will be a good contribution from contributors in neuropathology at MD Anderson, and also from the group at Cincinnati Children's, Mariam and the other groups. So this is going to be a very interesting paper on pediatric low-grade gliomas. In relevance to this, if we switch on to high-grade gliomas, even if we have a lot of exciting research going on, but the survival of the GBMs really hasn't been that, with less than 20% over 2 years. So in the pediatric high-grade gliomas, there's a group from MD Anderson who really focused on the epigenetics, the histone deacetylase inhibitors and how LSD1 inhibitors, etc. So it's going to be really step-by-step a nice review of that. And I think that would be a great thing back-to-back with the low-grade gliomas. It's a good complement. If we shift gears, another one of the tumors is DIPG, known as diffuse intrinsic pontine glioma, which has a median survival of only 12 months. As the global research continues to really find a breakthrough in the management of these patients, we really haven't gone much. But recently, we have found that the epigenetics is an important event. A lot of huff and puff is going on that. The group from the sick children and the group from the UCF has combined to really put this in shape a very interesting article. And I will log Dr. David Daniels because that's one of his areas of interest and expertise. If we move on to the embryonal tumors, which includes medullos, ATRTs, and other tumors like ETMR, actually myself and the other group from Dr. Gopalakrishnan's lab looked into the medulloblastoma. As you know, it is the most common malignant tumor and the most probably molecularly interrogated tumor in pediatrics. And so discussion about their 4 major molecular subtypes, the 12 subtypes of the subtypes, how each of these subtypes have so many epigenetic now important druggable targets, and how we are moved by able to get various drugs against that, and the clinical trials which are ongoing will be very nicely discussed in this. ATRT is a very malignant tumor, usually seen less than 5 years. I will have Dr. David Daniels talk about it because he's the senior author, and a group from sick children and also from Mayo has contributed to that. What is very exciting is the ependymoma. It's the third malignant tumor. And as we, over the last 7-8 years, we have been able to understand that there is a subgroup of ependymomas in younger kids, which basically we really don't know, and the survival is extremely poor. So I've been able to bring in two great actually contributors. One is Stephen Mack from TCH and Amy Smith, who is running the current ependymoma COG trial. So both of them really have nicely put forward. One is the clinical and one is the basic component with special focus on epigenetics. Finally, we have also the ETMR, which I mentioned is a subtype of these embryonal tumors, contributors being from the sick children front. They just, we were cleared yesterday, I think. They did the resubmission and we have sent it out for public. This is going to be very exciting because now we have the 3 subtypes of ETMR, and its relevance is also going to be spelled out in that manuscript. And finally, we have been able to get Martin from Germany, who has done a lot of work on choroid plexus tumors, especially the choroid plexus carcinomas, and its association with P53. So this is going to be very interesting, and the relevance has been well explained and how the molecularly they dissected. This is from the group from Germany, from the sick children URI, and from Alabama. So that is going to be interesting. And the last one is something which I thought is interesting because survivorship is interesting. We look into the survivorship. Is epigenetics really defines how the survivor is going to be for pediatric brain tumors? Nothing has really been done on that. So epigenetics and survivorship in pediatric brain tumors is going to be the last, one of the last contributors in this. And this is, this could be interesting because the Cincinnati group and Scott Pomeroy from Dana-Farber has contributed to this. So these are the 10 chapters which extensively goes across the spectrum of most of the diseases in pediatric brain tumors. And I think it's going to be a very nice summary and an interesting addition. And thanks to Dr. Sheehan once again for giving us the opportunity to put this forward. And from here, I will stop and ask Dr. Daniels to move ahead with the rest.
Thank you, Dr. Khatua. As you can see, he's presenting from Houston, which is in the middle of a storm, one of many storms this year. And obviously, I'm coming from Minnesota, and this is my virtual background, but this is the view right outside my window. This is part of the Mayo Medical School and the research building that I work in. And it looks exactly like this today, which is really odd for a September day, like a later September day like this, because it's usually already starting to get cold. So I'm gonna make this larger here.
Just while you work on that, I want to encourage people to use the chat feature. We can obviously respond in real time if people have any questions, and we can also monitor what's going on Instagram and YouTube for questions as well. So we'll try to make it as interactive as possible.
Yep, that sounds perfect. So I'm a pediatric neurosurgeon at the Mayo Clinic. For those that don't know me, I have a 50% research appointment, and I really study brain tumors. That's both in the operating room and in the laboratory. So I guess I'm very subspecialized, even more subspecialized than most, being a pediatric neurosurgeon and then just really doing brain tumors. I decided I didn't want to do shunts and Chiaris and all the other things, so I gave those to my partners to really just specialize in the brain tumors. I did my residency at the Mayo Clinic, and I subsequently finished a pediatric fellowship at St. Jude Children's Hospital, working with Rick Boop as one of the primary mentors. But I also have a research background with a PhD in medicinal chemistry. So really kind of rational drug design, drug delivery is my background in the lab. And then from a clinical standpoint, it is brain tumors. And so it's kind of a really niche type of practice that I've kind of carved out for myself at Mayo Clinic. Okay, so my lab studies 2 tumors, and as Dr. Khatua gave us a really good overview of the pediatric tumors, the ones that I study are highly malignant tumors that occur in children and are really simply bland genetic tumors, I think, as Dr. Khatua said, but result in a host of epigenetic changes that likely drive tumorigenesis. And, you know, when I was in graduate school in the late 90s, the word epigenetics was something that was just starting to be formed. And it was unclear even that cancer had an epigenetic component. And now we realize that that's a major component probably for all types of cancer to some degree. One of the prominent tumors that I study is the DIPG, the diffuse intrinsic pontine glioma. This is the brainstem glioma that lives in children. That's essentially a death sentence. This is every parent's worst nightmare to get this diagnosis. And that's really because we don't have anything effective to treat these children with. Yes, radiation therapy works and is palliative for a period of time, but ultimately all these children succumb to their disease. The average life expectancy is somewhere between 9 and 12 months afterwards. The staggering concept here is that there's been countless clinical trials, well over 100 clinical trials that have been performed with various drugs, and 0 have ever found any benefit. And so that's the part that's really quite challenging. The landscape for this tumor from a molecular standpoint was changed in 2012 when 2 groups independently found that there was a histone driver mutation in the majority of these lesions. So Susie Baker was one of them at St. Jude Children's Hospital, where she studied essentially just DIPG tumors, and they found a very high rate, about 70% have a histone mutation. There was a second study that was done by the Toronto Sick Kids group that was looking at high-grade gliomas, and a subset of them were DIPGs, but they found the same type of mutation. I can tell you that most people quote the fact that DIPG tumors have about a 70% chance of having this histone mutation based on that initial work, but it's much higher than that. The H3K27M mutation is the highest, or the one that occurs most, and there's 16 H3 genes. So you can see that there's probably additional H3 genes that can become mutated with different mutations, not just the methionine, that can actually lead to this. And so that's one of the main reasons why I would have to say that the majority of DIPG tumors all have a histone mutation. Based on this data, the WHO reclassified DIPG tumors, and now these are referred to as diffuse midline gliomas, histone mutated. And I think that's going to have to change a little bit as well, based on the fact that we're starting to find that there's more than just the H3K27M, et cetera. So why has countless clinical trials failed for this tumor? And one of the main reasons is likely that we're just not getting drugs into the tumor. Okay, so these tumors notoriously don't have a lot of enhancement compared to like a high-grade glioma in the supratentorial compartment that has enhancement. So the blood-brain barrier is probably a lot tighter for these DIPG tumors. And I guess one of the things that we notice when we biopsy them is that I would say over the majority of them, just histological grade probably ends up being a grade 2 or a grade 3 and not a grade 4 tumor. However, they are graded all as a grade 4 tumor if they have the histone mutation. They all behave bad, the outcomes are horrible. And so one of the main things is that we're probably just not getting, it's not that we don't have drugs that might be effective, we're just not getting the drugs to that location. And so there's numerous things that people are looking at across the country to enhance the delivery of drugs to these tumors. But one of them is on the forefront is convection enhanced delivery. And that's where we implant a catheter into the brainstem and then infuse drugs over a pressure gradient into the direct area. Okay, so Mark Swedane recently finished a phase 1 trial that was published last year that showed that this technique is safe in a larger cohort of children, and a phase 2 trial with his drug is starting to get underway. It isn't quite set up yet. That's with the radioactive iodine molecule, I-131, what they use for, I've seen used for neuroblastoma is in clinical trial. So it's a beta emitting particle, and it's also conjugated to a B7H3 where we know that these tumors have a high expression of that. And so that's how it hones to the tumor. There's other things about convection enhanced delivery, however, that haven't been explored. People have looked at the mechanical aspects of putting this under pressure, but it hasn't been realized as far as how do the drugs actually affect how they're being distributed with convection enhanced delivery. And so I have a graduate student in my lab who is working on this as part of her PhD thesis, and really looking at how does molecular weight affect the distribution. And these are some of the work that we've published on recently, showing that, and this makes sense, that as you increase the molecular weight of these compounds, you're going to decrease the volume of distribution, which sort of makes sense. But there's a mathematical relationship to this that's reproducible. Lipophilicity. How does drugs that are highly lipophilic affect their distribution when given via convection enhanced delivery? That's something that we're working on. And last but not least is, what about the efflux pumps? So, you know, if these compounds are getting efflux right out of the brainstem, they're obviously not going to be effective. And so these are the things we've done the molecular weight studies. We're halfway through the efflux pumps and lipophilicity, trying to characterize this. But this is going to come up with just kind of an overall summary mathematically of how these drugs distribute and how can we pick an optimal drug via convection enhanced delivery. Another route that we're looking at heavily is nanoparticle delivery, peptide guided delivery. And then there's ultrasound, focused ultrasound guided drug delivery as well. And so these are things that people are actively looking across the country. And I expect good things will ultimately come from this work. I think it's not just picking the right drug, but it's also picking the right way to deliver this drug that's going to hopefully make an important impact. As Dr. Khatua is saying, the special issue is really based on epigenetics. And I'd have to say that probably well over half of the amount of work that we do in our laboratory is epigenetic based. So DIPG is an epigenetically driven tumor. So there is global loss of H3K27 trimethylation. That's shown on this Western blot here. And so most tumors and other cells have a high expression of K27 trimethylation, but the K27M tumors do not. They have this global loss. So if they have the methionine mutation, it has this direct loss of K27. This is sometimes hard for people to understand because there's, again, 16 H3 genes. So if one of those genes has that methionine mutation, it causes the other genes then to lose their ability essentially to be trimethylated. Now that's oversimplifying things because things still have some basal trimethylation present. People have been focusing on that aspect. We've noticed that it's not just specific lysines and epigenetic changes, but there's also serines that get phosphorylated, specifically S28, which is right next to the lysine 27, and serine 10, which is next to an important lysine 9. And so we see that there's a whole host of epigenetic ripples that are going through this tumor that likely drive its tumorigenesis, specifically keeping the tumor in a more stem-like state, in a state that is going to not differentiate and propagate itself. So some of the tools that we've been developing have been isogenic lines, taking patient-derived cell lines and then reverting some of these specific mutations. For the DIPG work, it's taking the methionine mutation and making it back into lysine. And so this is a DIPG cell line from a patient then that we reverted back to lysine. And we can see when we do that, so it no longer has a methionine mutation, regain back that 27 trimethylation. And we also start to see changes in these other serine phosphorylations that we were looking at. And specifically what we found, and I don't have all the data shown here, but we show that when we do this revertant, the cell lines grow less, they grow, the tumors are less, grow less at less or slower speed, less ability to form the tumors and colonies than if they had that histone mutation present. We're also changing certain serine back into alanine, and so that can't be phosphorylated. And then there's also other ways to actually make this hyperphosphorylatable to take a look at how these epigenetic consequences go. Our lab recently got an R01 that is really centered on looking at these epigenetic changes that occur. So what I'm going to do now is kind of switch gears into ATRT, and this was specifically some of the work that went into this production and this special issue. This again was spearheaded by us at the Mayo Clinic. The first author is Cody Nesvik. He is a neurosurgery resident, but a research fellow in the lab. And then the folks from Toronto Sick Kids, Dr. Lefei Cousin and Dr. Annie Huang. And then one of our neuropathologists here at Mayo Clinic, Aditya Regnham. And really it was an overview of ATRT and then talking about some of the newer advances, but I'm also going to add some of the work that our lab is doing right now and trying to take advantage of some of these new advances and new concepts. So ATRT is atypical teratoid rhabdoid tumor. So it's of the rhabdoid tumor predeposition or variants. This is a 2-year-old child that presented with headaches, nausea, vomiting, but mostly just an increasing head circumference. And as you can see, there's a very large tumor in the left hemisphere that we resected. I probably see maybe one ATRT a year, so it is fairly a rare tumor compared to some of the other malignant brain tumors. But it's still common enough that we really need to come up with an answer because again, prognosis is quite dismal for these children. This case here is one that I remember most, and it was because I did this with Dr. Boop in Memphis. And it was one of those rare days that they saw snow in Memphis and the whole city shut down. And I distinctly remember that. Let me turn this phone off. These tumors have a predisposition for affecting very young kids in the age of 1, 2, maybe up to 3 years of age is the most common age group that we see this in. And it's quite a devastating tumor with overall poor prognosis. The defining thing about ATRT is it's very similar to DIPG in that it has essentially a genetic mutation, a very simple mutation that really drives its tumorigenesis through complex epigenetic mechanisms. So specifically, it's loss of SMARCB1 or INI1. This protein goes under a lot of different names, but this protein that is lost, it's biallelic loss. So that means that these tumors do not express any of this protein. It's part of the SWE/SNF chromatin remodeling complex. And that remodeling complex is needed for neuronal stem cell differentiation. And so you can see how if you have loss of that, then we have loss of stem cell differentiation, et cetera. And so the ATRT tumor has been known for a long period of time to have this loss of chromosome 22, which then we subsequently found out that it was this SMARCB1 or INI1 that's actually missing in these tumors. And what's really come out in the last decade is that there's subgroups, and Dr. Khatua is kind of getting to this, is there's subgroups within subgroups and subgroups within other subgroups. But for ATRT, we've been able to pull out 3 specific subgroups that kind of define this disease. This is based on DNA methylation profiling and RNA sequence profiling. And one of the common themes, so the 3 subtypes are MYC, sonic hedgehog, and tyrosinase. And they kind of occur in different age groups, as the tyrosinase is seen more in newborns and infants, whereas the sonic hedgehog in toddlers, where the older children tend to have more of the MYC subtype. But again, from an epigenetic standpoint, what we see is there's global loss of H3K27 acetylation. So that lysine on 27 is known to be affected in DIPG by having a methionine group substituted, which then leads to this loss of trimethylation. In ATRT, what we see is an actual opposite, is a loss of acetylation. So acetylation we know is an activator of gene transcription in a chromatin sense, if it's present. And whereas if it's trimethylated, that is more of gene repression and not transcription. And so there's a difference in essentially the effects that this type of epigenetic changes can lead to. And so it's only been apparent over the past handful of years about these genetic or epigenetic, I should say, partly driven subtypes for ATRT. And so the question is, can we develop targeted therapy for this? And that work is ongoing. One of the things that our lab has done, again, using the same type of principles and tools that we're doing for DIPG is, let's just flip the phenotype where we have loss of INI1 or SMARCB1. Let's regain that back and see what it does. And so these are patient-derived tumor cell lines. These come from other groups. These are not our own. And when we restore SMARCB1 and give that back, these cells stop proliferating. They stop forming colonies. They're no longer able to make tumors. And so we have a lot of work, and this is the stuff that's being done by Cody Nesvik, the neurosurgery resident in our lab, who is studying this in more detail, trying to further hone in on this and really kind of define the epigenetic changes that are occurring with this. I should point out that I think this is really interesting, will probably bore everybody else. This upper panel here, TE, is these typical enhancers, and this SE is super enhancers. In ATRT, when we lose that SMARCB1, we see a loss of those typical enhancers that drive cell differentiation. But what we see is a continued gain or a continued function of things that drive proliferation. And I guess that makes sense, is that the cells still want to proliferate and grow, but then they can't differentiate. And that goes along with some of the phenotypes that we're seeing. So I think that's all that I kind of prepared for some of the specific science and kind of an overview of these tumors. I'm going to hand it back here to Dr. Khatua. I want to again thank Dr. Sheenahan, Dr. D'Amico, and everybody else for listening and having us.
Thank you, man. That was great. Really cutting edge work going on. It's incredible. And, it brings us to this next generation of what we're able to do as, physician scientists and really basic science researchers, where we're able to tailor medicine to the disease itself and learn as much about it as possible. And I think that's phenomenal work. Dr. Khatua, did you have more to add?
So, no, I had a question because I know you're from that world, So we have in MD Anderson, a large adult medulloblastoma. A surgeon wanted to do that. So we had about 200 medulloblastoma patients over the last 20 years. My question to you in the adult world is, I know like the sonic hedge, these are common in this adult and the group. How do you target these? What's the difference if a sonic hedge subclass comes to you versus we know what's ours? Do you have subclassification of sonic hedge? And the reason I'm asking is any sonic hedge adult who walks into your clinic, usually, I don't know what's the molecular subtypes, but we routinely test for P53. So in your world, can you tell us what is the difference between adults and pediatric medulloblastomas?
Is this to Randy, I assume? Or is this to me?
This is to Randy. Yeah, I mean, Randy and Jason, I mean, all that adult, anything or even anybody.
Sorry, I actually didn't realize you were talking to me.
Oh, okay.
But you were, one more time, I'm sorry if you could repeat it.
So what I'm saying is,
I apologize. I'm in the OR right now. I'm listening to what's going on.
So is it okay?
Yeah, absolutely.
So it's just a question, So we know we have a lot of pediatric sonic hedge, which contributes about 30-40%. In your adults, it's much common. But the thing is, whenever a pediatric patient comes in with a sonic hedge subtype, we immediately look for P53 and all those subtypes. What is the difference in targeting these adults with sonic hedge versus ours? Do you know?
I don't know. Yeah, I don't know. I think that realistically, we have to, replicate what Dr. Daniels has done in the pediatric population and bring it to adult tumors as well. I think what, and this just goes to my own practice because, there's in New York City, we have 5 academic centers. We don't get every tumor. And at Lenox Hill, we don't have a pediatrics department. It's all done at Cohen's. But what's very curious to me and what's I think spectacular about the work you guys do is these are extremely rare tumors. I mean, for you to do this work on both medullo and ATRTs, how are you getting these patients? Are they being referred in for research? I mean, what's the volume like there that this is possible even?
Yeah. Well, so from our stuff, I can tell you, I probably have the largest collection of Dipg, and I'm trying to assemble the largest collection of ATRT material. So what does that mean? That means all the material that I get at Mayo Clinic, which is some, but again, it's not going to be that numbers that you're talking about. So I'm on email, cold emailing people up. Hey, I'm Dr. Dave Daniels from Mayo Clinic. I see that you published on this. I see that you have this. Can we have that? And so I literally, anytime any of this type of material hits, that I know of hits, a published manuscript, this or that, we're trying to get those, that material. As far as cell lines, people are easily happy, to give that. As far as getting the actual patient samples, that becomes harder. And so there's a Dipg registry, which I'm very thankful for. They've, rewrite a proposal to them and they review it. And then they've given us samples and to augment. It's also me contacting people like at MD Anderson, at Phoenix Children's Hospital, it's like, Dave Adelson, what do you got, for these type of samples and stuff. So it's me panhandling, trying to get all that tissue and stuff. And so that is the most challenging part. I can say that my the research fellow, the Cody, has countless, infinite amount of emails that he sent out to people that, you know, includes my name on it. That just says, hey, we're trying to get this stuff. So that's it.
Well, I think I mean, both you, Dr. Daniels, and Dr. Khatua, and certainly Randy and I, we work at specialized centers that do catch, in some instances, we're, you know, in my state alone, we're catching patients, you know, 6, 8 hours drive from where we are, and that catchment area does get some of these rare tumors, but it also, I think, speaks to the nimbleness and the courage and the resourcefulness of the neuro-oncology community to pull together our research activities and to try to enhance outcomes in very, very challenging disease states that are oftentimes extraordinarily rare. And I mean, I've seen this in my practice, and I know you've all of us have seen it in our respective practices where you'll see a tumor and work in an environment where you've got, fortunately, colleagues who you can bounce cases off of and then pull together research across institutions and even across countries in many instances. And I think that speaks to the support that we have at the Journal of Neuro-Oncology too. And we have 9 different societies partnering around the journal, and the work that you both have contributed to, I think, will spur more interest in pediatric neuro-oncology. But Soumen, I think you've asked a challenging question about the adult versus pediatrics. It's complex, but certainly P53 mutational studies are going to be commonplace in adult brain tumor workup. But I think that we don't have the answers that sufficient answers to, at least from the literature that I would know, in answer to your question. I think we need to do more work. But I really appreciate both of you and contributing to this, and Soumen particularly. The credit belongs to you and your contributors, not to me. And I will say that in this special issue, unlike some issues that are put together by some journals, it's never an economic driver. It's not a cost. This is the Journal of Neuro-Oncology putting this special issue together to benefit the field. There's no cost to contributors. There's no cost to editors to put this together. But it was a high value, high important special issue. Thank you so much for leading it and featuring it today.
Yeah, guys, thank you so much. We try to limit these to 30 minutes or so. We're about 5 minutes over. I think the future of pediatric neuro-oncology and treatment of these things is very bright because of people like you. And I think that, like Dr. Sheehan said, very important issue. So it's our pleasure to have you today. I'll send you guys a link where this will be posted. Feel free to share it. Hopefully we can get Dr. Daniel some more collaborators. And once they see his data, and we'll be sure to promote it as well. So tune in next week. We got another great episode. Let me tell you who exactly is coming on board.
Thank you.
We have Dr. Raza will be here next week talking about, I believe, skull-based tumors. So we'll have a great time. So thank you guys so much.
Thank you.
See you all next week. Take care.
Take care. Bye-bye.